Hello, this is David, and the purpose of this installment is to express and share ideas about the placenta. And this is not really an educational video. It's more a video to express ideas and see what people think and see if I could engender further conversation. So, the topic that I'm interested in is intrauterine growth restriction and how it manifests in the placenta. And part of my information is based on a partial reading of the writing of people like Peter Kaufman and more recently Dr. John Kingdom. And my understanding of a critical point that they made is they were looking at placentas in IUGR babies, especially severe early onset. And they noticed that these placentas tend to be small. And ultimately they came up with this idea of branching versus non-branching angiogenesis. Branching versus non-branching angiogenesis. And I don't want to do too much injustice to their idea. But basically my understanding is that the placenta has these sort of, stem villi that then branch out into various distal villi and sort of like a branching structure. And then at some point in the tree, you have a structure that is an immature intermediate villus. Immature intermediate villus. Immature intermediate. And so let's take this villus. And it's an immature intermediate villus. And it's growing along. And then it has several options. One is it could bifurcate, and the bifurcation could then bifurcate, and that could bifurcate. And all of these will still be immature intermediate villi. Or the immature intermediate villus could say, you know what? I no longer want to contribute to the bifurcation. I want to become a mature intermediate villus, which this is like an edematous looking structure, the immature intermediate villus. A mature intermediate villus is thinner, more heavily collagenized, and then it starts to do something. It starts to sprout with these syncytiotrophoblastic knots, and that's why their literature sort of deals with what's a true sprout and what's not a true knot and what's a true sprout. But then it starts to engender these terminal villi. And both the mature intermediate villi and these terminal exchange villi are areas that are sufficiently capillarized by the fetus to allow for gas exchange with the mother, where these immature intermediate villi aren't really, but they allow for more dichotomous branching. So the mature intermediate villus allows for elaboration of distal villus sprouting. The immature intermediate villi allows for bifurcation and further growth. So that's concept one. And the other concept that they sort of worked in is that the kind of branching that you get, or the kind of angiogenesis that you get as you continue the dichotomous branching of these immature immature intermediate villi, is what they call branching angiogenesis. Whereas in these mature villi is sort of like this form of non-branching, non-bifurcating angiogenesis, and that's what gives rise to the terminal circulatory units. And so, mathematically, if we examine a placenta, and when I sort of do this, it sort of harkens to, if any of you guys know Vi Hart's sort of mathematical installments that are available through the Khan Academy, and she's very funny, and she'll say things like, she'll say, suppose you're doodling in math class. Let's say the math teacher is very boring. And we all did this when we were kids. We would draw a line, and then we would, each time our line hits one of the horizontal lines on the page, we would have to split our line up. And then now we've hit another line, so this splits, and this has to split, and this has to split, etc., etc., etc. And so this structure rapidly becomes a fractal. And so, because it has this kind of self-symmetry, there's sort of like this constant iteration, this replication of the pattern that you use in order to determine where you replicate. And that reminds me a lot of, so with the magic of modern computer science, I've redrawn this sort of schematic of a branching placenta. So imagine let's say this is like your umbilical cord, let's just say for the sake of argument, and then these could be your first chorionic vessel branches, and then it plunges into the placental disc and it bifurcates, and imagine that's your stem villus, and that gives rise to generation, and that gives rise to generation, and then at some point you get sort of these sort of intermediate villi that have a dichotomous branching potential that we'll call immature, but at some point the placenta will need to upscale. The needs of the fetus will get greater, and will sort of outstrip the capacity of this sort of branching structure that's not particularly adapted to gas exchange, and it'll say, hey, I need more oxygen. So it'll take one of these guys out of circulation, and then he'll make some sprouts, and he'll create some terminal villi. But the trade-off is that now this branch of the tree is gone. He's not going to have any more children. Whereas, let's say this point here, he could still branch again, and this one could still branch again, and this one could still branch again, but this one can't. So this one is now taken out. So you could imagine that the earlier the placenta starts to need to do that, the greater you restrict the overall ultimate growth potential of the placenta. So imagine, for the sake of argument, because once you become a mature intermediate villus, you could no longer undergo this kind of dichotomous branching. You could only elaborate terminal villi. So to take an extreme example, imagine the placenta is really starved early. Imagine this villus had to become a mature intermediate villus. You would lose all of this. In other words, you would lose 1/4 of the total growth potential of the placenta. So it seems that throughout development, you have your fetus, and your fetus's metabolic needs are scaling up. And the placenta is trying to keep up, but the placenta is also trying to grow. And if the fetus is early on not putting too much of a demand, you could have a lot of branching, but some villi, usually from the more distal portion, are going to get taken out of circulation and turned into terminal, whereas others will continue as branching structures. But if the pressures are such that the fetus is feeling an early pressure, early inability of the placenta to scale to meet the metabolic needs of the fetus, then early on you're going to make too many conversions into non-branching structures, and you're going to restrict the overall ultimate growth potential of the placenta. So this is why I think they argued that early onset IUGR is associated with an ultimately small placenta. And that brings to the so one mathematical concept that I think needs to be developed, and it should be relatively straightforward, is to develop a mathematical model where you have this sort of fractal type dichotomous branching, and then you model what happens as you not randomly, but as you start to take terminal branches out of circulation and or as you take intermediate villi, immature intermediate villi out of circulation and make them into exchange units that now could beget these sort of terminal gas exchange villi but could no longer undergo dichotomous branching. And so mathematically model how that'll affect the scaling of this structure. And I think some modeling for that could be done. And I think there's one other concept that needs to be taken into account. So if we imagine the fetus, so let's think of the fetus as a sphere. So a sphere's surface area upscales by x to the power of 2, but its volume upscales by x to the power of 3. So if as this sphere goes from here to here to here, its volume increases by a factor of the radius to the power of 3. So it's a form of geometric growth. So you can imagine here it is. But the placenta, which is sort of growing as a fractal structure, I would imagine that its ability to scale up is sort of something like this, and so that there's an obligatory intersection point, and hopefully labor or term will be around here. And so, but I think that there's a biological problem in that this point of metabolic intersection, where the scaling capacity of the placenta intersects the metabolic scaling of the fetus, where that intersects, that point, which is where the placenta could no longer keep up, is not perfectly coupled to labor. is not perfectly coupled to the onset of labor. And that may be a cause of intrauterine fetal demise, and that's quite important. And I think that's actually an important concept, and I think that's almost like one of the complexities, or you might say flaws, in our fetal design, is that this point of intersection is not perfectly coupled. But the capacity of the placenta to scale up is probably related in some ways to how much it's restricted its overall growth potential early on. So we know the growth parameters of the baby, we know what the ratios of placenta to fetus are in various cases. I think using this concept of scaling, and also using the concept of this branching versus non-branching angiogenesis, I would propose that we could do some really interesting mathematical modeling, and then we could potentially predict points at which small placentas in severe early onset IUGR, if labor doesn't happen, they will lead to intrauterine fetal demise, which would be a point of intersection. So that's one suggestion that I would have, one prospectus, or one thing I sort of put out an appeal to all of you guys. Anybody who's listening, if you want to sort of investigate these ideas and try some mathematical modeling, I strongly suspect that this could be fruitful. The concept of the scaling was developed in some papers by Dr. Salafia, by Dr. Carolyn Salafia, wrote some, I believe, hopefully I'm spelling that name right. She's really one of the best mathematical thinkers that I could find in the literature on placenta, and she's done some of this work on scaling, and really is the person that introduced me to the notion of scaling. And there's even a concept in evolutionary biology called allometric scaling. So if you want to read a little bit on how metabolic needs scale up, and it's sort of an interesting concept that I won't get into now. And then the concept of the fractal growth of the placenta, I don't really have any formal training of it. Some of it is loosely based, believe it or not, on the video that Vi Hart puts out on the internet, and especially her some inspiration, although not direct ideation, comes from some of the stuff that she wrote about or presented about plants, because the branching of although that was more related to Fibonacci numbers, but still the branching of the placenta does remind me very much of the branching of a tree, both poetically and metaphorically, as well as mathematically. And there is some interesting mathematical work on the scaling of a tree. So what I would urge people to do is take Kaufman and idea of branching versus non-branching angiogenesis, and imagine how making an exchange unit, a mature intermediate villus, will suddenly compromise the overall growth capacity, and then try to mathematically model how that will influence the ultimate point of intersection where fetal metabolic needs intersect with placental needs. And that's, well, let's call this my video clip on placental theoretical ideation. And so that ends this first installment.